Hi, everyone. Thanks for joining us today for this episode of QBI TV. And it's called Looking Beyond DNA to See Cancer with New Clarity. Now, this is a joint presentation between the Quantitative Biosciences Institute, QBI, which fosters collaborations across the biomedical and physical sciences, seeking quantitative methods to address pressing problems in biology and biomedicine. And it's motivated by problems of human disease. And QBI is committed to investigating fundamental biological mechanisms because ultimately, solutions to many diseases have been revealed by unexpected discoveries in the basic sciences. So my name is Elizabeth Blackburn. I'm Professor Emerita at UCSF, and I'm Vice Chair of the Stand Up to Cancer Advisory Committee. So what's Stand Up to Cancer? Its mission is to raise funds to accelerate the pace of groundbreaking translational research in order to get new therapies to patients quickly and to save lives. And like QBI, Stand Up to Cancer is inherently collaborative in nature, and we're going to touch on that during the session with respect to QBI. Just a little bit more about Stand Up to Cancer. Since 2009, over $700 million have been pledged to support Stand Up to Cancer's innovative research programs. Over 200 clinical trials have been launched by Stand Up to Cancer investigators involving over 2,000 cancer scientists in the cancer research community, over 19,000 patients participating in trials. So there have been other collaborative nature enterprises of Stand Up to Cancer, launching a series of convergence grants, and these bring together physical and computational scientists, basic scientists, oncologists. So today, we're going to highlight some important link by researchers in the Cancer Cell Map Initiative, CCMI. Now, this was founded in 2015 between scientists at QBI at UCSF and at UC San Diego. And so we're very lucky today to have on our panel for today the CCMI co-directors, Nevan Krogan, who's the director of QBI at UCSF, and Trey Idaker of UCSD. And in a minute, I will ask both of these panelists each to introduce themselves. Now, we're going to be talking about cancer, and we do need to point out to our listening and participating audience, we aren't clinicians, so just please to keep that in mind. And so we do hope we will get questions from the audience, and we very much look forward to your participation in this discussion. And so I'm going to now switch to introducing, or asking, in fact, Nevan and Trey to to introduce themselves. So first of all, let's start. Nevan, would you like to introduce yourself?
Sure. Thank you, Liz, and thank you, Liz, for moderating this discussion here today. So I'm Nevan Krogan. I'm a professor and director of the Quantitative Biosciences Institute at UCSF, and my research focuses on the development of various tools that we use in integrative ways to study a variety of different disease areas. And the foundation of the CCMI is really combining of these different tools that we're going to be talking about today in collaboration with tools and approaches at UCSD. And it's in a close collaboration with Trey Idaker, who I'll let now introduce himself.
Trey, please go ahead.
Hi, thanks, Liz. Yeah, so I'm the co-director of CCMI together with Nevan. I'm a professor of medicine in the Division of Genetics at UC San Diego. So CCMI was really conceived of originally as a north-south California axis between two University of California campuses. My research is similar to Nevan's in the way he described it. So we are at the center of work in genetics, genomics, and bioinformatics, and especially some of the sort of exciting discoveries that have been going on in artificial intelligence and how those might be applied to patient care and analyze cancer basic research and clinical data.
Great. So I think we should just launch into our discussion. And particularly, I think I'd love to hear just a little bit more about how did you start CCMI?
Sure. Maybe I could start and then Trey could chime in. So part of the motivation behind the formation of CCMI, the Cancer Cell Map Initiative, was a realization that there was a limit in what scientists could extract simply from genomic information. As many of us are aware of, there's been large sequencing efforts focused on a variety of disease areas, in particular cancer. And we've got this set of genes now that have been linked to cancer. And we realized this list of genes is not by itself leading us to the answers. Also the realization that there's a lot of heterogeneity with respect to the mutations. You have this long tail of mutations that people talk about, and you have people with the same cancers, they're sequencing their tumors, and there's only an overlap of a couple genes from patient to patient. And the question is there an overlap here at the end of the day? And the answer is yes, there is an overlap, but not at the gene level. To see this overlap and to interpret this genomic data, we got to get to the protein level. So I would argue that the CCMI is a big focus of it is just simply looking at the functional units of the cell, the proteins, and we study how the proteins physically and functionally interact. And Trey and I have been working together for many years to generate these maps in various contexts. And we focused our attention now over the last 6-7 years on cancer. And the generation of these maps, it's incredibly powerful. I would argue that it helps you interpret the genomic information. It helps you collapse down the different sets of genes into interpretable pathways and complexes. And what can these maps do? Well, number one, they can be very predictive with respect to prognosis. That we've shown that in the past in the CCMI. Number two, it allows you to be more precise with respect to precision medicine. If you have a set of mutations, you can put the mutations on the map connected to a cancer, and we can be predictive about which treatment should be most effective. And number three, personally, this is the thing that I'm most excited about, a little bit longer term, but it provides a more global view of the biology. It opens up new avenues. It identifies new genes, new proteins, new complexes, new pathways connected to a particular cancer. And then that opens up novel therapeutic avenues as well. So I think that these maps that we're generating, and we're bringing in a lot of different scientists to generate these maps and interpret these maps, are incredibly powerful. And the CCMI, and led by Trey and I, we just published several papers a couple of weeks ago, which I think very much highlighted the power of what we can do. And we're just at the infancy here, but what's possible with respect to these approaches that we are pioneering in the Cancer Cell Map Initiative.
Yeah, the exciting goal and success, I think, of the CCMI is you've gone beyond genes to map protein-protein interactions, these PPI maps, as you call them in the papers. And this has critical goals, and it's just such a natural partnership with Stand Up to Cancer, because the goal is to improve prognosis and discover new targets and therapeutic directions. And so, Trey, I'd like to hear your perspective too, very much, coming as you do with an additional layer and depth of computational approaches to this.
Well, sure, Liz. I think Nevan actually said it very, very well. So I'll have just a bit to add. But I think one complementary way I could approach this question is, if you think about human genomics, and the Genome Project, and you think about the timeline, 2001 or so, we had the first human genomes appear, and that was largely due to the success of the Human Genome Project and all of that technology. Call it genetic DNA technology, and that was now 20 years ago, right? Almost exactly 20 years ago when those genomes appeared. What's happened in the intervening time? Well, we saw a decade of sequencing tumors in a very exciting way, and there were projects such as the Cancer Genome Atlas that have now sequenced hundreds, if not thousands, of examples of different tumor types. Breast cancer alone has several thousand representative genomes that any researcher can access if they're interested in what are the patterns of mutations that cause that disease, for instance. So all of this, oh, thank you. See, there's wonderful graphics we have as we talk here. So all of this work, which we are not controlling, just so the audience knows, all of this work is an important foundation for what Nevan just talked about. What we're now proposing to do is to connect the individual gene mutations people find in tumors. And as he said, the biggest finding of all of these sequencing efforts really is that everyone is different. Just like if I compare Liz's genome to my genome to Nevan's genome, of course, all of our genomes are snowflakes. The genome is what makes us who we are, as it were. Is it any real surprise that the same is true of our cancers as we have them? And it's absolutely true, which is part of now the challenge. So where the state of the field is we can sequence a patient's tumor fairly rapidly, fairly inexpensively, and of course those the speed and cost of sequencing are getting better all the time. But now how do we really make sense of it? And you're only going to be able to make sense of this disease, as people are finding out, by having the map of, in a sense, the rest of cell biology and tissue biology of the disease. These genes don't act alone. They act to create circuits. And you're seeing here a circuit diagram that we've mapped for breast cancer. Before, during Nevan's answer, you were seeing another map we've constructed for sort of spanning multiple tumor types. And these maps are what really allow you to connect the dots in a given patient's disease and see how their tumor is actually similar to another person's tumor, even though they're mutating different genes, those two tumors, but they're mutating the same circuit. And on that's the basis by which you can treat cancer.
And what was so exciting was that going beyond genes, as you did in this way, by looking at all the interacting partners among these proteins, what you started to see was, yes, mutations start to show up in not what was thought to be, the common, ones that everyone thought of, TP53, but things that interact with these. And so those mutations also now, by interacting in the network, they make sense of what's driving the cancers. So you start to see convergences into not the huge, as you said, everyone's a snowflake, but it's starting to make sense. It's coalescing into different pathways.
Absolutely. And if I can just interject, Liz, there's one other sort of important prior work that we're standing on the shoulders of. If you're thinking about all research stands on the shoulders of previous giants, and I mentioned the Human Genome Project and the Cancer Genome Atlas Project is absolutely a foundation for what we're doing. The other is sort of this important idea that has long been in the cancer field, that there are these hallmarks of cancer, these pathways that onto which all these mutations converge. So in a sense, it's not a novel idea that cancer is a disease of these circuits or pathways of multiple genes. That idea has been out there a long time. I think the novelty is, how do we apply a systematic approach to map these pathways, just like systematic approaches were so successful at mapping the genome in the first place?
We have already a question from one of the participants, which I think really draws further into this question of, well, what are we going to do about this? And particularly, how can we talk about different contexts? For example, different cancers. So your papers, dealt with head and neck cancer and with breast cancer, and that dives straight into what can we do and what was learned. So would Nevan, would you like to?
Sure, I could start that as you referred to.
How this started to inform the very real situations: breast cancer, head and neck cancer. Why was this important to do?
So we started our approach, as you refer to, on breast cancer and head and neck cancer. And honestly, we could have focused on any cancer. The main reason we did that was because we had these great collaborations here in San Francisco and San Diego. We had Silvio Gutkind in San Diego and Jennifer Grandis in San Francisco, experts on head and neck cancer. And then on breast cancer, we had Alan Ashworth here, Laura Esterman, and Laura Vanfier. So we're really leveraging this great expertise both in San Diego and San Francisco. And that's a big part of the Cancer Cell Map initiative, is collaboration. Collaboration is key for this initiative to be successful. Now we're going to be expanding to other cancer types. And there's great commonality as you go from cancer to cancer, which I think is a great strength. But we're still not done with breast cancer and head and neck cancer. And there's different ways you can use these mapping approaches to get at insight behind these cancers. So for example, you can look at these maps like we've done in these papers in the presence of cancerous cells versus non-cancerous cells. And there's certain interactions that exist in cancerous cells that you don't see in the healthy cells. We argue these represent fantastic therapeutic targets. So that's one axis. Another axis that we've highlighted here in these papers is that we can take one gene that's highly mutated and look at these maps in the presence of the individual mutations. We did this for PIK3CA. This image here is representing that particular work. So we can look at what effects individual point mutations have on the networks. That can be incredibly revealing with respect to directions in precision medicine and identifying new targets. And then a third area, which actually we're just starting to do, which I'm actually very excited about, is looking at these maps in the presence of cells that are resistant to drugs. So trying to understand drug resistance. This is a big problem with cancer treatment. And what we've done here, although it wasn't in these particular papers, what we're looking at here, we're making cells resistant to particular drugs. So cetuximab is a first-in-line drug for head and neck cancer. The problem is you get resistance very quickly. So we made cells resistant to cetuximab, and then we generated those maps in those cetuximab-resistant cells compared to cells that were sensitive to cetuximab. And to me, it was quite remarkable to see the rewiring with respect to these maps. And it's in these rewiring of these maps, I think, has great insight with respect to the underlying biology behind resistance, which then will help inform polypharmacy. Because that's one of the approaches that we need to get to, is which combinations of drugs would be powerful. And that's where then now AI approaches that Trey was talking about before are going to become particularly relevant. So we can use these approaches across different cancers, but we can take one cancer and go very deep, looking at that cancer in a number of different ways, all of which is very revealing.
And it just occurs to me from what you just said in this example that you can look at what rewiring produced, and that'll probably eventually start to tell you how is that rewiring done, so you could attack the rewiring before the cancer could even have a chance to get to the point where now it's rewired itself, and now you have to come in with another whack-a-mole, therapeutic. And so I think this is very exciting to be able to actually start to understand what even goes on to rewire. And now, Trey, what do you think? You also, in these papers, in science papers, covered breast cancer example. Is there anything particularly from that example that struck you, in addition to what Nevan said? Is there a specific situation that struck you that nobody had ever realized in breast cancer before?
Oh, you're muted, Trey.
Thank you. So I think one underlying piece of biology that comes up in all of these papers is this what's called the PIK3CA AKT-mTOR pathway, our axis, which is activated or modulated in some way in many different tumor types and in many individual patient tumors. And so the surprising connection that you see across all three of these papers, including the breast cancer paper, is the role of PIK3CA, and as Nevan said, how mutations in PIK3CA interact differently depending on, of course, where those mutations are with the biology of the tumor and how it progresses. Perfect graphic for what I'm talking about. And a lot of that work, as Nevan mentioned, was validated in the lab of Silvio Gutkind, both for head and neck and again for breast cancer. So maybe that's enough said, Liz. But I think, there's, in terms of the circuits that are defined by these maps, two things. You like to find things that are already known, okay? And certainly it was known that PIK3CA modulated these different cancer types. What was less known is how exactly different point mutations in proteins like PIK3CA do that job, right? So again, big picture, not a surprise that generally this pathway is important, or that PIK3CA is important, but exactly how particular events in particular tumors come about is now much better known due to these maps. So that's sort of things about cancer we already knew, and now we know a bit better or a lot better, depending on the case. There's also cases where brand new complexes are discovered in the data that simply weren't appreciated, involving proteins that weren't well appreciated in cancer at all. And so one example that comes out not just for one cancer type, but actually in many of the cancer types we looked at, for solid tumors at least, is the example of collagen. So collagen complexes are well known to form the dominant component of what's called the extracellular matrix, or the ECM. It's through that extracellular matrix that tumors must expand, proliferate, and if you're very unlucky, metastasize. So it's logical that ECM has a big role to play in tumor progression. But what had never been noticed before is that the collagen complexes themselves are under heavy mutation pressure in many cancer types. And the reason that had never been noticed was because people had only looked one gene at a time. And when you simply look one gene at a time, you don't see any one of these collagen encoding genes that's particularly remarkable. Okay, some of them almost were noticed in the prior studies. They were sort of flagged as interesting, but weren't focused on. But when you look at the protein complex level, like Nevan's been talking about, and you sort of connect the dots and realize there's 20 family members that come together and usually triplets to form these complexes that then form these fibers of collagen. Now you have the bigger picture, and you can see that in fact you're looking at the convergence, as you said, of rare mutations across this gene family.
Yeah, very exciting, because you're really finally much more going towards the functional entities of this really complex set of systems. And speaking of complexity, we have a question from the one of the participants who just asks, and I forgot to ask you, are you analyzing tissues or single cells, both? How do you look at that, and what's technically feasible at the moment? What would you like to see?
Maybe I could start. So the maps that we're generating right now, in the cancerous cells, the non-cancerous cells, are cell lines, or immortalized cell lines, both with breast cancer and with head and neck cancer and other cancers now. But we're making predictions from these cell lines and then going to potentially more relevant systems to validate those predictions. Okay, so that's like a mouse model, like we was just shown when Trey was speaking. Working with Silvio Gutkind in San Diego. We have great clinical samples, clinical connections, both here in San Francisco and in San Diego. We can test these predictions with clinical information, like at the I-SPY trial that's ongoing here, that's being led by Laura Vantier and Laura Esserman. And we can also get clinical samples, where we can go and do more targeted predictions, say from tissues, like Mark is alluding to here in his question. Now, we haven't done anything here with respect to single cells. You can do single cell RNA-seq at present time to do single cell protein-protein interaction mapping. It's not feasible. Probably in the future, with these new mass specs that are coming online, they are getting to single cell, not with respect to protein-protein interactions, but profiling. So it'll be interesting to see what we can extract once we get to a single cell level with respect to these networks. But there's a ton of information we're getting just simply looking at these more tractable cell lines, generating these maps in disease states, non-disease states, getting these amazing predictions, and then testing them in more relevant system, and then a reiterative way going back and altering how we generate these maps.
And Liz, if I can chime in here, I think one thing for sure that neither Nevan nor I want to communicate is that we've solved this problem, or this is the ultimate map. These maps you're seeing flashed up on the screen are the ultimate answer to cancer. We think they're a very promising approach, where just like the first human genome that was sequenced, that laid down sort of a paradigm. And now that you have the paradigm, then you want to let it rip over the next decade. Okay, and so that's somewhere on that curve, I think, is where the CCMI is now, and probably at the very beginning stages of that growth curve. So we need to figure out how to do this mapping in single cells. Absolutely. We need to figure out how to do the mapping better, faster, cheaper, in different, so we can scan quickly across not only different tissue types, but individual tumors of that, and get real representation. So far, we've looked at essentially, arguably, Nevan, we've looked at 3 to 6 examples of each of these tumor types, and even that's up for discussion. Whereas with genome sequencing at this point in time, there's now hundreds of examples that have been viewed, and so that's clearly the direction this project is going to go, as well as the technology gets faster and the approaches become vetted. So I think it's an exciting time. We are certainly at the beginning of this time.
And I'd like to bring up just a comparison to the Genome project that Trey talked about earlier. Once the Genome project was done, we checked the box and said, all right, we're done. This is it. But the genome, the DNA is, obviously relatively static compared to proteins. proteins are not static. They're interacting with other proteins, in different contexts. So what you'd really want are maps across many different cell types, plus and minus disease, plus and minus stresses, plus and minus different mutations. So it's not just like, okay, now we're done this map like the genome was done. This is going to go on for a long time. But that's a realization that the biology is complex. The proteins are the functional units, and they're very complicated. So on one hand, it's like, oh, we got a lot of work to do. On the other hand, hey, we got a lot of work to do. We're going to be in business for a long time generating these maps. So to me, that's very exciting.
But you're not just generating maps. I mean, you're seeing the dynamics, as you alluded to.
That's right.
You can see rewiring happening as a cancer progresses into, for example, resistance to a particular therapeutic treatment, or as it goes into a metastatic state and so forth. You can see the dynamics because it's the networks. And I think that's what's so exciting, that, the whole networks functionally are changing in this dynamic.
And you're referring to the next frontier, in many ways, temporal aspect of these maps, as you say, right? As you introduce mutations, as cancer progresses, as you get drugs involved, right? So this is, we're laying a static map here, but now what we're going to do is to go to the temporal aspect of that. And that's a very exciting component of this.
Now, you alluded to, interacting with the with actual clinical samples too. And this brings me to another thing that I think has been such a kind of exciting part of QBI and, of course, this CCMI, and that is the collaborative nature of it. And that, of course, is very close to the DNA, if you'll excuse the word, of the Stand Up to Cancer, which, was founded on the idea that, in fact, if people got together from different institutions and so on, and instead of competing with each other directly, actually collaborated, you could move things faster in cancer, especially to benefit patients. So collaboration, as we all appreciate so much better now than I think we did just a few years ago even, it is really key. And we've already seen just in CCMI collaboration from very different kinds of expertises, from the very clinical oncology through understanding cell and molecular biology, protein chemistry, computational approaches. Just love to hear a little bit more about your thoughts about what makes collaborations work best in your hands. Everybody talks about it, but you live and breathe it. I'd love to hear the perspectives from both of you, because, it's one of these things in science. We always want to do science better. What do we learn by people listening to people like you folks who've done it so successfully?
I'll let Trey start.
Sure, I can start with that one. And in fact, I'm going to answer one of the questions that we've gotten here in the process and kill two birds with one stone in a way. So start with an example. So there's different kinds of collaboration. One important form of collaboration that institute or an idea like the CCMI can really catalyze is you recruit budding new scientists to the effort who read your papers. Read, for instance, this perspective piece launching the CCMI that Nevan and I and a few others published back in 2015. So Hannah Carter, who at the time was a postdoc in Rachel Karchin's lab at Johns Hopkins, read that piece, applied to my lab, came for a very brief postdoc before she became a professor herself. And in the process, to answer Mark Schumann's question, published a series of really nice work, I think. One with me on it, and mostly with me not on it. Now that she's out of my lab with in her own research program on this exact question of how do germline polymorphisms, that is to say those that you're born with, interact with the acquired so-called somatic mutations that are then pushing those tumors, as you said, Liz, down the road, the whack-a-mole, you have this sort of germline state that then predisposes you to certain moles, that is to say somatic mutations that arise, and then further mutations during cancer progression. So Hannah's entire career has essentially developed. So she was attracted by the CCMI. She came to UCSD. She now set up her own lab at UCSD, and it's all about the question. And for instance, in my lab, she had this beautiful result where she showed that back to the PIK3CA AKT mTOR pathway, you have a variety of germline variants you're born with. and that affect that pathway. Some rev the engine of that pathway. So this is a proliferative pathway. And so some I think of as sort of, you're not idling the engine. The engine goes at maybe 1500 RPM, not 500 RPM in these people. And so this pathway is primed for growth. And so then in those people that have these certain mutations in various proteins that rev that pathway up, now later in life, you tend to get mutations, further mutations in that pathway that are required somatically. So this kind of now gives us a complete picture of how the germline primes the pathway for later tumorigenesis. And then since that time, Hannah's done beautiful work on her own, focusing on MHC and the germline state of MHC in terms of what cancer peptides later are displayed and either detected and destroyed or allowed to pass through. And it's just a fascinating body of work. All that comes back to an attraction of Hannah to the CCMI piece and work, of a number of our labs in the area. So anyway, that's one form of collaboration is in setting a vision, I think, of a project that attracts the best minds to the problem. Just quickly, and I know I've been talking a long time, and I'll pass.
I love that perspective.
Yeah. And then just quickly, I think the other key, and then I'll pass it to Nevan, is realizing, not having the hubris that you can do it. I mean, you always, on the one hand, who better to do something than yourself? If you want something done, do it yourself. But you have to know your limits, and you have to know when to collaborate and who to collaborate with. You, of course, in our modern world, want to reach out and get the best and brightest out there, the experts in the very thing that you're not good at, right?
Maybe what I would add here to Trey's comment, as Trey was speaking, I was thinking back actually when Trey and I first met. I was a student at the University of Toronto. He was an assistant professor at UCSD. Although it may not look like it, he's older than me, just to put that on the record. And we started to interact, and Trey was coming from a background more of a computational background. I was coming from more experimental. And I think we realized that we both brought something to the table. So during the last, 16, 17 years since we first met, this is obviously a good example, I think, of collaboration. We've learned a lot from Trey on the computational front. A lot of the tools we have in our lab are based on Trey's work. And I think Trey's learned a lot from our experimental approaches. And to me, that's really that collaborative spirit is what bore the Cancer Cell Map initiative. And now we have groups. It's not just proteomics, it's genetics, it's structural biology, it's chemical biology, it's connection to the clinic. And for me personally, I don't want to work on projects that only one lab can do at the end of the day, right? And here's a great picture of a number of the CCMI participants, and each one of them has a very distinct and tool that or expertise that they bring to the table, and we can get so much more done when we work together. And I think we saw this during the pandemic, obviously, how fast science can move, really out of fear, I would say, when people work together. And now that hopefully as the dust settling on the pandemic, we're now going to refocus on other disease areas like cancer, and let's apply what we learned during that pandemic, and let's move as fast as we can. Let's move so much faster, realizing the value that collaboration has. So I know it's been a tragedy, but I'm so excited now to take what we've learned during the pandemic and focus it on diseases like cancer, because I think 20 years from now, we're going to have treatments for cancer that we wouldn't have had if we didn't have the pandemic. And a lot of it is because we learned how to work together more effectively.
And to quickly answer this, a question that's on our slide, on our screen right now, I think that's, Senzo makes a perfect additional point for why collaboration is key, because there's no way you're going to tackle mapping the cancer protein network, even much less the rest of cancer cell biology you're going to need to know. There's no way you're going to do that with just even two labs represented here on the screen, or three, including Liz. You're going to need to have a large buy-in from the community, just like systematic projects that again created foundations for this one, such as the Human Genome Project, followed by the Cancer Genome Atlas projects. Those, of course, involved armies of people, and it was the only way to get it done.
those armies, there's about 20 groups highlighted here, and they're they're leaders of research groups. How do you think the actual total number of folks adds up to in for CCMI?
Well, we have a number of PIs represented here, and in each lab,
that's only
many people. So we're talking about hundreds of different scientists just in San Francisco and in San Diego. But now what we're doing is branching out and making connections in other institutions, not just in the United States, but around the world as well. And I think these 3 papers that we've been talking about nicely lay the foundation and illustrate the power of collaboration. And they're getting a lot of interest, and people are coming to say, hey, we want to get on board. We want to get on board this collaborative CCMI train. So I think we're in a great position now to kind of take this to the next level with respect to collaborations. And maybe just one other thing I'd add along those lines. So we started the Cancer Cell Map Initiative, as Trey was saying, back in, I think, 2015 or 2014, 2015. But since then, we've used that blueprint to start other mapping initiatives on other disease areas. So we have the Psychiatric Cell Map Initiative, focused on different neuropsychiatric disorders like autism and schizophrenia. We have the Host Pathogen Mapping Initiative, focused on various pathogens, including SARS-CoV-2. And we're having just as much success in these other areas. And a couple points to make about these different cell mapping initiatives. The technology is very, very similar. All right. So it's the same technology, just different set of genes, different cell type. The technology is similar, and that's exciting. What's even more exciting is that the biology is overlapping. So it's the same genes being mutated in cancer that are being hijacked by SARS-CoV-2. It's the same genes being mutated in autism that are being hijacked by Zika. And to me, this makes sense. You have these Achilles heels of the cell, and they become mutated and result in disease X, Y, or Z, or a virus has evolved to attack these important genes and complexes and pathways. So in a way, we're not just making connections between different genes and proteins, we're making connections between the siloed scientific community that is each focused on their different disease areas. So we're bringing together scientists across these different disease areas, and I'm really excited about the potential of doing that in terms of maybe there's a treatment for one disease that'd be better for another disease, or at least there's knowledge and reagents that could be brought to bear in another disease. So it's not just about the genes and proteins that we're making connections is that we're making it with people, not just in the cancer world, but across many different disease areas.
And from the computational side, Trey, what do you think in terms of those approaches that have been, so successful and so promising as well in cancer, as you've been describing? But where do you see the challenges and particular opportunities in other diseases as well as cancer when you take it from a computational perspective?
Sure, Liz, thanks for the question. So let's talk about AI in cancer and other diseases. And another foundational aspect here is we are living in a time when it's hardly a day goes by that you don't read about some new success of deep learning. Whether it's deep learning has beat the Jeopardy. Well, I guess back then it was earlier forms of AI, but whether it's deep learning has beat the Go champion, or we've recently seen how it potentially has solved the protein folding problem with the AlphaFold application. I think deep learning is a very scarily general procedure for how one can learn patterns in data and how they associate with outcomes. And I've said it, I've chosen my words so that we can perhaps now map that onto all sorts of problems in basic and especially clinical cancer research. And so, I think before in the green room, so to speak, Liz, you and I were talking about different challenges that are essentially predictive challenges in cancer clinical medicine, which are well, at least on the surface of it, well addressed by advances in areas of AI like deep learning. And so you had brought up the challenge of predictive, or I should say preventative cancer medicine. So if we can understand whether or not a tumor is likely to progress, given the current assessment of a patient, and that assessment could be, of course, molecular and genetic, but that assessment also includes lots of clinical data that are collected during visits and follow-ups. To what extent can you then look at all those data and now predict the likelihood of the tumor further developing? That's certainly one big challenge. I'm going to skip a bunch, and then skipping over a number of challenges in between. If the cancer does progress, now you're in a clinical situation where you want a treatment. And so there again, you want to prescribe what is the best treatment or treatments, what's the best regimen, as it were, given the input data that the tumor presents and the patient presents with. The CCMI has had a train of papers focused on that particular challenge, which is called sort of drug response prediction. And the exciting thing about how the CCMI has approached it is we do it using the kinds of maps we've been talking about here. So we can take maps of protein-protein interactions among cancer proteins and use those to inform decision-making in the clinic using deep learning. So I think it's a really exciting time. Now, to cycle back to your original question of how does all of this bear on other diseases, just like Nevan said, we have a core set of technologies here, experimental for mapping these networks and pathways, also computational. And these same core technologies that are useful for predicting how a given set of cancer mutations that's created a cancer is likely to respond to drug A, B, and C. Okay, that same technology is certainly promising for application to other genetic diseases. And Nevan mentioned PCMI, the Psychiatric Cell Mapping Initiative. There, a poster disease for us is autism, because autism is created by a combination of rare, so-called de novo and common germline variants in a very complex way, or much like people think cancer arises.
Yeah, I think this is really fantastically exciting because, you said AI scarily, but actually, perhaps we should be scared by the fact that there are so many diseases that are still so challenging. We're excited about the advances in cancer, and yet it remains a huge, fearful disease. And, so there are certain things about the potential to use all of these technologies, combining in deep learning with all of the deep understanding of cell networks and interacting functional pathways, and saying, yeah, we could maybe make it a less scary disease if we use these things. So, and then to that point, I think we have a nice question here, which is that we've talked very much at a pretty technical level, but everybody, one way or another, is affected by cancer. There isn't a human being who doesn't know another human being or have a family member who has cancer pretty much on the planet. And so, and the numbers remain, extraordinary. Every 2 or 3 people will experience cancer. 1 out of every 2 or 3 people will experience cancer at some point in their lifetime. So we all care. And how are you going about conveying what you do to, as the questioner said, the lay or average person? What do you do? What are the communication routes that you each use?
I think this is an important point. We get the most of our funding from the government, and that's taxpayer money. And I think there's an obligation on us to inform the public on what we're doing and why we're doing it, and hopefully why it'll ultimately have an impact on the general public. And at the Quantitative Biosciences Institute at QBI, we're doing very creative things to try to get the word out. For example, we have these the salon. Here's a picture of a salon. We've partnered with a company where we bring together people from the public, and there's Alan Ashworth sitting there getting with a discussion around cancer research with the public. So this is one of the things that we do. There's efforts like A Taste for the Cure, and Laura Esserman here in San Francisco runs this, and this is where cooking combined with informing the general public around our cancer research. We merge this together. There's actually an event, ironically, next Monday night at the San Jose Sharks game, where they're combining cancer research and hockey, where we're going there we have a booth, I think. We're presenting our efforts to obviously to the hockey population about what we're doing around cancer. And here's another event that we had, another salon, a picture of Trey and I, along with Laura Vantier in Washington, D.C., where we had a discussion about, with the general public, that they were invited around our cancer research. So, we're doing creative things and things like this. And QBI TV is doing very creative things in terms of getting the word out on our research, not just on cancer, but other disease areas as well. So I think that's a very great question. And we're actually open to suggestions on how to improve getting the word out to the public. But we're trying in a variety of different creative ways.
Well, I can see again this natural partnership, why Stand Up to Cancer and QBI are such natural partners, because one of the big missions of Stand Up to Cancer has been, it was founded by entertainment industry leaders. Their whole job is communicating with the public, right? And so they've set out to generate awareness and educate the public on, cancer prevention as well as treatment opportunities, helping people diagnosed with cancer, become long-term survivors, not patients who don't survive their disease. And so I think this is such a key point for the reasons that you talk about, not only funding that allows this important work to happen, but for people to understand that there are, let's say there's hope. There's often treatments that actually might be applicable to their situation. And so I think that this is very exciting what you're saying. And Trey, would you like to add to it? And then I think we may be out of time. Wrap up. So Trey, please, you.
No, I'll make it quick, Liz. So just quickly, I would just add of the things Nevan mentioned. I would also add we have, I think, in both cities a great relationship with the Science Museum. Certainly down here, we have a great ongoing relationship with the Fleet Science Center in Balboa Park, where we give talks. And I believe you guys have a similar situation up there in SF. And then I would add, I think it's not exactly the question of outreach to the lay public, but certainly outreach to the clinic, right? I mean, outreach to the cancer patients themselves on the level of understanding what we're doing, but of course, importantly, on the level of their treatment. And so there, it might be important maybe just to close with a brief discussion of what we've done so far. And I think we need to do much more to get these ideas into the clinic. But importantly, a number of clinicians are quite engaged with the CCMI, both inside and outside of that list of faculty that was presented on the screen before. Down here, we've made nice progress with the so-called molecular tumor board at our cancer center. And so for people who don't know about these things, the very difficult cases of cancer are almost always discussed by a so-called tumor board, where you get like minds and actually diverse minds into a room to discuss what should be the best therapeutic plan for this patient. How do we get our results into these boards? And the answer is through the AI. So our results are encapsulated in these decision-making systems, which then sit alongside the doctors and patient advocates who participate in these councils. And I think that's one important piece of progress that we're beginning to have. Laura Vantier is also quite involved with organizing the I-SPY breast cancer clinical trial, and that's another important direct connection. And the way that she's designed I-SPY, in fact, allows for late-breaking biomarkers as come out of protein interaction maps into patient decision-making. So again, lots to talk about there, and we're out of time, but I think an important aspect of outreach is the clinic.
Yes. That's a great point to finish on. And you touched on one more thing, which is how important it is that we bring diverse minds into this whole very challenging and exciting era. And I just want to say thank you so much for being panelists on this episode. And I'm sure there will be many more exciting updates to hear. And congratulations on the terrific work that you're doing.
Thank you, Liz.
Thanks very much, Liz. That's great.
And thanks to Stand Up to Cancer as well for co-sponsoring this. Thanks everybody.
Bye, everyone.